Welcome to this lecture of biomathematics. We have been discussing differential calculus, basically how to calculate derivatives of different kinds of functions. So, we learn the idea of derivative and calculating them for a few functions. We will continue with this today and learn a couple of examples from biology. So, today we will briefly discuss a bit more about derivatives, the continuation of where we stopped last time, and at the end we will discuss two examples from biology where the idea of derivative is directly applied. So, let us start. So, the title of today's lecture is differentiation and its application in biology, and last time we learned a formula, actually two formulae, as you see in the screen here, that d by dx of x power n is equal to n x power n minus 1. This is the first formula that we learned. Then we learned the second formula, which is when you calculate the derivative of sum of two functions, f of x and g of x are two functions of x, and if you have the sum of them, f of x plus g of x, and find the derivative of them, derivative of the sum, the answer is basically you can find derivative of these individual functions and sum them. So, this is the second formula that we learned, and there is one more important formula that one should understand, one should learn, so that you can do the derivative or you can differentiate any function, pretty much any functions. So, this is the third formula that we are going to learn, and this is very interesting formula. So, have a look at the screen, and so this is called product rule. When you say product rule, it's like a product of two functions, f of x and g of x are two functions, and the product of this two function is f of x times g of x. So, this into this is times. So, f of x times g of x, so the product of two functions, and if you want to find the derivative of this product, d by dx of f of x times g of x, is the idea is very simple. First, find the derivative of f of x and multiply with g of x, and find the sum it with finding the derivative of g of x and multiplying with f of x. So, this is simple. First, find the derivative of f of x df by dx multiplied by g of x plus f of x into dg by dx. So, this is the product rule of differentiation. If you understand this, you can learn a lot of things just by extending this, just by using this just one formula. If you remember this just one formula, you can keep using this and learn find out derivatives of many functions actually. So, let us take the simplest examples for this formula. So, let us look the formula once more. The formula says f of x times g of x is a product, and its derivative d by dx of f of x times g of x is equal to derivative of the first function df by dx times the second function g of x plus the first function f of x times multiplied with the derivative of the second function, which is dg by dx. So, this is the formula, and now let us take the simplest example here. So, the simplest example is f of x is x itself, g of x is x itself. So, x times x is x square. So, last time if you remember, we learned the derivative of x square. So, we are going to calculate d by dx of x times x. That is, we are going to that basically nothing but d by dx of x square. That is what we are going to calculate. D by dx of x square, this is what exactly we, and we learned from the last time, we had this rule of this rule which we just said, that d by dx of x power n is n x power n minus 1. Using this rule for n equal to 2, it is clear that the derivative of this is d by dx of x square is 2x, n is 2, x power n minus 1, 2 minus 1 is 1, is 2x. So, from what we learned last time, we already know the answer that derivative of x square is 2x. Now, let us apply today's rule. the rule we learned is d by dx of f of x times g of x is df by dx into g of x plus f into dg by dx. So, this is what we learned. Now, let us apply this. F is, so in our example, in today's example, the first example we took is f of x is x and g of x is x. So, this is x into x. So, now let us say, so d, so basically we are going to find out d by dx of x into x. Now, df by dx, f is x. So, this is f is x. So, dx by dx is the first term and g is also x plus f is x, g is also x. This is what we get. So, now what is dx by dx? So, we all know what is dx by dx. It is nothing but 1. So, we know that dx by dx is 1 and this is 1. So, this is 1 times x plus 1 times x. So, let us look at the screen. So, d by dx of x times x is dx by x into x plus x into dx by x. There is no magic here. I just applied this formula. Once you apply this formula here, once I took this formula and applied this to this. So, you get this, if I apply this formula here, I get dx by x into x plus x into dx by dx, and noting that dx by dx is 1, we get 1 times x plus x times 1, which is 2x. So, we get the same answer. So, basically this is basically x plus x equal to 2x. This is something, 2x is the answer, same answer we got here. So, basically by just doing this, we get the answer we know. You can continue doing this for any other function, any function that we learned, and do it many times, and you will see that you can, just by knowing dx by dx is 1 and this rule, you can calculate derivative of any function. And at the maximum you will have to apply the sum rule we learned in some particular cases. So, if you know the sum rule and the product rule and the derivative of x power n, you can calculate derivative of any function. So, let us apply this product rule to some other cases. So, let us apply the product rule to some cases. So, now let us take the derivative of a constant times x. So, according to the product rule, you have two functions a and two things a and x, the product of a and x. So, the derivative of this will be dA by dx times x plus a times dx by dx. So, take the first one, derivative of the first one multiplied by the second, the x, plus keep the second one a constant and find the derivative of this, which is dx by dx. Now, a is a constant. So, in our case a is a constant, it is like example 2. Let us say a is just 2, then d 2 does not change with x, 2 is just a constant. If you, so for dA by if a is a constant, change in a is basically 0, a cannot change because it is just a constant. So, dA by dx change in a is like 0. So, if this is 0, this term becomes 0, then your answer is just a times dx by dx, which is 1. So, the answer is a. So, d by dx of a x is just a. So, this is something which we can learn just by applying our product rule. You can extend this, you can apply something else, you can say d by dx of a is another, a is constant, x square. This is nothing but you can, as you know, is you can write dA by dx into x square plus a times d by dx of x square. As I said, a is a constant, so derivative of a is 0. So, this term is 0. This is 0 times anything is 0. So, the first one is 0 plus a d by dx of x square is 2x. So, this is a times 2x. So, it's basically 2 a x. So, d by dx of x a x square is 2 times a times x. You can keep applying this formula to any function you like, and you will see that you can learn interesting things. So, let us just take one more example, some more example, we will come back to this. But another thing that we learned last time is e power x. So, let us quickly remember what we learned. We know that e power x can be written as the function e power x can be written as 1 plus x square by 2 plus x cube by 6 and so on and so forth. And the derivative of e power x is e power x itself. So, this is something that we learned last time, the derivative of e power x is e power x itself. Just revisit what we learned last time using this product rule. Like, so, just go back as an exercise, just go back to the lecture of slides of last lectures, the last lecture in fact, when we found the derivative of e power x, and try using the product rule, it will help you. So, let us think about it. If you want, let us say the function is 1 plus, so let us just quickly redo this. So, we said e power x is 1 plus x square by 2 plus x cube by 6. So, now d by dx of this is d by dx of e power x is nothing but d by dx of this whole thing, which is 1 plus x square by 2 plus x cube by 6. Now, as the sum rule says that if you want to find the derivative of the sum, you can take the derivative of each of these individual terms and sum them. So, d by dx, so the first one is d1 by dx, which is 0. Second one is dx by dx. Third one is d by dx of x square by 2, and this one is d by dx of x cube by 6. This is what it is. Now, the first, this here it is 1, this is 1. So, d1 by dx is 0, because 1 is a constant, just like we did a, 1 is a constant, change in 1 is 0, because there is 1 cannot change. Now, the second function, let us look at here, dx by dx is 1. So, this is 1, derivative of dx x is 1 plus, now we want to calculate derivative of x square by 2. So, now here we can use the product rule. So, x square by 2 is basically d by dx of half into x square. So, half is like a we had, half is a constant times x square. Then you can use again d by dx of 1 by 6 times x cube. 1 by 6 is a constant. So, 1 by 6 times x cube. So, you can use this product rule like d by dx of a times x square and d by dx of a times x cube, and you will see that finally you will get the answer e power x itself. So, each of by using the idea of infinite series, you will get that e power x itself. So, just as an exercise, just redo this what we learned last time. Okay, so, we also learned last time, we also mentioned that derivative of sin x and cos x. So, what is derivative of sin x and cos x? So, we learned that, look at this, we learned that sin x is x minus x cube by 6 plus x power 5 by 120 minus x power 7 by 50 40 and all that, and cos x is 1 minus x square by 2 plus x power 4 by 24 minus x power 6 by 720 and so on and so forth. So, derivative of sin x, if you use the this 3 rule which we learned, the d by dx of x power n, the first formula that we learned, use the second formula which is d by dx of sum of few functions, and use the third one d by dx of product of functions. If you use this formula to this sin x and calculate the derivative, you will get cos x. Just try doing this, and if you do that for cos x, you will get it as minus sin x. So, I will not do this in this, but just like we did e power x last time, you could just continue doing this, just try doing this and you will get the answer. So, now let us today try and understand what does this mean. What does it mean to say that derivative of sin x is cos x? What does this mean? So, first let us look at this graphically. So, let us in the next slide we will look at the plot of sin x and cos x. So, have a look at here. So, what is plotted in red is sin x. At x equal to 0, it is 0, and when x is equal to pi by 2, 3.14 is pi, this is around 3.14, this is pi, this is pi by 2, which is 90 degrees. So, when x is 90 degree, d by dx, the cos sin x is basically 1, sin 90 is 1, and sin 180, which is pi by sin, this is 3.14, which is pi. So, sin pi, which is 180 is 0, sin 3 pi by 2, which is between pi and 2 pi is 3 pi by 2, which is minus 1, and again sin 2 pi is 0, and you can see that the red curve represents sin and the blue curve represent cos of x. So, look at it for 0, cos 0 is 1. So, starting from 1, cos pi is 0, again cos 2 pi is 1. So, cos pi is minus 1, cos 2 pi is plus 1, and cos pi by 2 is 0. So, you can see that the blue curve represents the cos of x and the red curve represents sin of x. Now, what our claim is that we claim the derivative of sin x is cos x. We claim that. We also claim that derivative means slope. We learn that derivative means slope. So, let us think about this. So, what does it mean that if you calculate the slope here at this local point here, you will get the slope as 1. And you can see that this is, look at, concentrate only on the red curve. So, just look at only the red curve. So, when you look at the red curve, you can see that this is a positive slope. What does it mean? So, if you look at the red curve, if you just look at the red curve has something like this. So, this is x and this is y. As x increases, y is also increasing. So, you can see this, the red curve is something like this. So, in this part here, this part, as x increases, y increases. So, dy by dx, which is as x changes, does the y change or not? If y also increases, x changes, dy dx is greater than 0, positive. What does it mean? There is a positive slope, the slope is greater than 0. So, that is what it means. So, now have a look at, just plot this and see whenever x and y are increasing, the slope is positive. So, this is a rule for even for a, so you might have seen that for a straight line which looks like this, the slope is positive, the slope is positive slope. You might have also seen that if the straight line looks like this. Here this is x and this is y. As the x increases, y is decreasing. For this value of x, for this particular value of x, this is y value. For this particular value x, this is y value. So, x increase from here to here, but the y decrease from here to here. So, as you can see, this has a negative slope. So, let me just make this once more clear. Positive slope means as x increases, y also increases. Negative slope means as x increases, y decreases. Now, if you look at this part of the sin x curve, as x increases, y also increases, the slope is positive. So, let us look at this curve here. For the red curve, before this particular value of pi by 2, the slope is positive. That means, the cos x value, which is the slope or which is the derivative, is positive. And here, if you look at this part, just that is after pi by 2 and before pi, the red curve has a negative slope. It is decreasing as the x increases, the function is decreasing. So, it has a negative slope. So, that means its derivative has to be negative. So, see here, the cosine, the cos is negative here. So, the cos was positive here, negative here, and then it is negative. Similarly, you will see that, so whenever you have an positive slope and negative slope, the function will increase and decrease. And you should also note that the peak, the point which changes from negative slope to positive slope, positive slope to negative slope, the slope is 0. At this peak, the slope turns out to be 0. Even at this peak, slope is 0. At this peak, slope is 0. So, this is an interesting phenomenon which you will soon learn, that whenever there is a peak of any function, if you find the slope or the derivative of that peak, you will get that value 0. This is something that you will quickly learn, we will discuss soon, but just note that when you plot sin x and find the derivative, you will get 0 at these peaks where the slope is changing, and also now it has positive slopes and negative slope. So, this is the graphical representation of sin x and cos x. We will come back to this, but just plot it in your computer, play with it, and just also if you have a graph sheet, just plot sin x and cos x and just play with it and see when the x is increasing, where is y increasing, when x is decreasing, where is y decreasing, and what is the slope at each point, so on and so forth. If you just play around, you will get a better feeling. So, once you have a feeling for functions, once you understand this function in a graphical representation, once you understand the graphical representation of the function and you get some idea about the slope, where is slope increasing, where is slope decreasing, where is slope changing, where is slope 0, that will help you a lot in understanding the calculus. So, if you want to understand mathematics very well, it is important that you play with graphs. So, you take pen and paper or graph sheet and just keep plotting. That is one way by which you can learn a lot of mathematics. Okay, so we now learned a few functions. We learned all polynomial like x, x square, x cube, x power 4 functions. We also learned the derivative of these functions and the sum of their functions like e power x, which is sum of x square, x cube, x power 4, etc. Even sin x and cos x can be written as some linear combinations of x power n's. So, we learn the derivative of sin x and cos x also. Using this technique, you can learn derivative of pretty much any function that we discussed so far. So, as an exercise, you could go ahead and learn the derivative of all the functions that we discussed in the previous lectures, and then see what you get, and you will see that it is things that you thought was very difficult turns out to be very easy by just learning this just 3 formulas. Those 3 formulas that we discussed, that derivative of x power n, the sum rule and the product rule. Okay, so now that we understand simple functions and their derivatives, let us learn 2 applications in biology, which will be basically 2 simple applications, and using which we will use whatever we learned, some things that we learned about derivatives and functions, and that will help us to understand a few things in this to examine the biological example that we are going to discuss. So, the first application in biology, the first example I am going to take is polymerization of filaments, for example, growth of actin filaments. So, as you might know, there are many filaments in biology. Actin is just one example. So, for those who are not familiar with actin, let me quickly tell what actin filaments are. Actin is a cytoskeletal polymer. It polymerizes in the cytoskeleton of a polymer. It is a protein that polymerizes and becomes long filaments. So, just like any polymer, it is made of many monomer units. So, let us quickly represent here. So, for simplicity, I will represent monomer by this blocks. So, this is monomer of actin. So, monomer of actin, actin is a, so monomer of actin is called G actin. So, G actin, it is called globule actin sometime. So, the monomer of actin is G actin, and this monomer polymerizes and becomes a long polymer. So, polymerizes means what? They join together. So, one after another, many monomers join together and form a long polymer. So, we are going to understand this process of polymerization. Filament, this G actin becomes F actin. So, this is called F actin, because this is filamentous actin, it is a filament. So, this G actin becomes F actin. We are going to learn something about, we are going to use the idea of derivative, the idea of calculus and functions we learned to understand something about this process. So, it turns out that lot of things in biology like cell motility, the way cells scroll, the force generation for cells to scroll, lot of things depends on this polymerization reaction and its rate and so on and so forth. Not just actin, there are other filaments like microtubules. Microtubule is another important cytoskeletal filaments. They are also, the monomers join together and form long polymers, long filaments. So, the monomer of microtubule is called tubulin. So, whatever we are going to discuss now is in general polymerization of any polymer, for example, actin, or it could be microtubule, or it could be any other filament. There are examples in bacteria also like FTSZ, PAR-M. These are some examples of filaments seen in bacteria, a bacterial cytoskeleton. So, this filaments they polymerize, and we want to learn something about the polymerization and how fast they polymer and so on and so forth. So, let us just quickly look at this slide here, this diagram here. So, in this cartoon here, as I discussed, this single box here represents G actin, which is the monomer of actin, and this is a filament. And with some rate Kp, this monomer binds onto this filament and elongates. So, the Kp is the rate of polymerization. Kp is typically something per second, let us say 10 per second, 20 per second. So, that is Kp. Kp is the rate of polymerization. What does it mean to say Kp is equal to 10 per second? What does it? It means that in 1 second, this 10 of this will be added. That is the meaning of saying Kp equal to 10 per second. And Kd, which is the depolymerization rate, which is also something per second, let us say 2 per second. 2 per second means in 1 second, 2 of them will depolymerize, 2 monomers. This one and this one can depolymerize on an average in every second. That is what the meaning of Kd equal to 2 per second. So, Kp and Kd are 2 rates. Kp is the polymerization rate. It is a rate with which this filaments polymer. So, this monomers, they just keep joining together and becoming longer and long filament. And Kd is the rate of depolymerization. If you have this monomer, they depolymerize and come off. So, this rate is Kd. So, the net polymerization obviously is what we want to calculate, but before that, when you think about this, polymerization, for this polymerization to happen, all these monomers, the G actin monomers, has to come and bind here. So, it is clear that the more the G actin monomers, the more likely that they will bind, because there will be all around there will be monomers. So, these monomers will come and hit this polymer and likely that they will bind. So, the rate of binding is directly proportional to C, which is the concentration of unpolymerized G monomer actin concentration in the solution. So, if you think about it, if you think about this, so if you think about an experimental setup, you have a container, let us say cuvette. In this cuvette, you will have some small seed of polymer, and there will be many monomers, like there will be a concentration. So, there will be many monomers floating all around. So, C is basically the concentration of this monomers, concentration. This is concentration means number by volume. In this volume, how many monomers are there? That is the concentration. So, the more they are, the more likely that they will go and hit and then form this binding. So, the Kp, the rate of polymerization, is likely to be more. The more this monomers are, more likely that they will bind. So, this is proportional to this C, directly proportional to the C, and this proportionality constant is some rate K0. So, the rate K0 here is the proportionality constant. So, the Kp, the polymerization rate, is K0 times the concentration C. So, once we realize this, we can think about what is the net speed with which this will polymerize, the net speed of filament growth, because the growth of filament is important in biology, because the faster the filament grows, the more force this actin can apply on the cell walls and cell membranes, and so that the cells can crawl. So, cell motility depends crucially on the rate of polymerization and so on. So, now let us look at here. Once we understand that Kp is K0 times C, the rate of polymerization Kp is K0 C, we can write down the growth speed is Kp minus Kd, which is common sense again. The net speed is whatever polymerized minus whatever depolymerized. So, if you have 10 monomers polymerizing per second and 2 monomers depolymerizing in second, the net speed of the filament will be 8 monomers increasing, the polymerizing. The length of this filament will increase by factor 8 monomers. So, the growth speed is nothing but Kp minus Kd, the polymerization rate minus the depolymerization rate. And we saw that the polymerization rate is K0 times C. So, I can substitute this here. So, the V is nothing but K0 times C minus Kd. So, we get an equation for the speed, growth speed or the growth velocity I should say. So, v represents for the speed or velocity. So, the growth velocity is k 0 times c minus k d. So, this is the function. So, we have a function. So, let us look at v, which is a function of c, is k 0 times c minus k d. In other words, you can write this is your f of x, the function velocity is a function of concentration. So, the function we are interested in here is velocity. So, this is like f of x equal to m x plus c. You might have seen this equation. As we learned, this is a straight line. So, we can plot this. So, let us imagine some experiment that you are doing. So, let us say you want to calculate the velocity. So, let us say you are doing an experiment where you have a setup, you have a cuvette using fluorescent microscopy. You can look at the length of the actin by varying the concentration, that is, you fix the monomer concentration of actin to a particular value, let us say 1 micromolar, 2 micromolar, 0.5 micromolar. So, you keep a constant value of the free G actin monomer concentration and may look at how fast this filament is growing. And if you do that, you can get a plot, you can make a graph which is like this. So, have a look at this graph. So, this point, the red points here is for different values of concentration, 0 concentration. So, basically let us start from the right end, 1 micromolar. So, the x axis is concentration, y axis is the velocity, x axis in micromolar, concentration unit is micromolar, molar is the unit of concentration. So, micromolar means 10 power 6 molar. This is the typical range with which one can do experiments on actin monomer growth, actin polymerization. So, and then you can look at the velocity. So, velocity is plotted here in subunit per second. How many subunits grew per second? In other words, monomer per second I should say. So, later when the concentration is 1 micromolar, the velocity is just below 10, it is actually 9.5. When it is 0.8, some value here, again 0.7, 0.6, 0.5, 0.4, 0.3, 0.2, somewhere between 0.2 and 0.1, this is crossing the value of 0. So, you can extend this. So, if you plot this much, you can join them and draw a straight line and extend this till you hit the y axis. So, this is one experiment, then this one curve you can plot by doing the experiment. By varying the concentration and measuring the velocity, one can get the data points and plot this curve. So, this is a very interesting experiment done in 1980s by two biologists. So, one person who did is M.F. Collier. So, you can look at the papers of M.F. Collier, or you can look at the papers of T. Pollard. These are two scientists who did experiments on actin polymerization. So, look at the papers of Collier and Pollard. So, go to Google Scholar, which is basically scholar.google.com. Then search for M.F. Collier or T. Pollard and look at the papers in 1980s, late 70s and 80s. So, late 1970s, early 1980s. So, roughly around this time, if you look at the papers, you can see that they have done experiments studying actin polymerization, exactly the same experiment that we discussed. And they wanted to find out what is the rate of polymerization, what is the rate of depolymerization, how fast they polymerize and how fast they depolymerize. So, they basically did this, they varied the concentration of free G actin monomers and measured how fast the polymer grows. So, they could look at and plot velocity versus concentration, and let us look what can they do. So, if they get such a curve, from this we know that the slope of such a curve, so the y axis, if you just calculate the slope here, okay, slope is dV by dC, change in concentration. When you change the concentration by some amount, how much the velocity changes. So, dV by dC, and that will give you the k0, which is the intrinsic rate of polymerization. K0 is the intrinsic rate of polymerization, and you multiply with the concentration, you get the rate of Kp, rate of polymerization. So, this is the rate constant, and what happens when C equal to 0? When C equal to 0, V is, this term of 0 is just minus Kd. So, the value at which, at 0 concentration, whatever be the value, that will be the minus Kd, but you can't do experiment at 0 concentration, right. So, what can you do? You can do experiments at large concentrations in this positive, this part, and extend this and extrapolate it and plot here, and then look at the value at which that hits the y axis, that is called y intercept, and you will see that this value is Kd. The value at 0 concentration, the value at 0 concentration is Kd. So, you can see that V is minus Kd, negative, below 0. So, that is this value, and you will also see that this is the value, the concentration at which polymerization equal to depolymerization. So, this is the critical concentration. So, you might know that the critical concentration is the concentration at which polymerization equal to depolymerization, that is Kp equal to Kd. So, Kp equal to Kd is polymerization rate equal to depolymerization rate. So, if you plot a curve which is C versus V, at some particular point velocity will be 0. So, this is 0. So, the point, the concentration at which the velocity is 0, velocity 0 means Kp equal to Kd, and we know that Kp, we learn that Kp is nothing but K0 C is equal to Kd. So, C is the concentration. So, the concentration at which these are equal, this would imply that the concentration at which they are equal is Kd by K0. So, this is known as the critical concentration. Kd by K0 will give you the critical concentration. So, you can do two things, either you can find out Kd and critical concentration, and then from that you can calculate K0, or you can calculate the slope and calculate K0, and you can calculate Kd, and if you know Kd and K0, you can also calculate the critical concentration. So, just by plotting and understanding that the slope or the derivative is nothing but the polymerization rate constant, we are essentially using calculus, differential calculus, the simplest form of differential calculus to learn something about the experiment. Now, once you understand this much, so what did we learn? What are the bottom lines? So, we learned one thing. So, we learned, the bottom line is velocity is K0 times C minus Kd, and dV by dC, that is the change in velocity when you change the concentration, is nothing but the slope of this curve. This is nothing but the slope of this curve, and slope is nothing but K0. So, this is C and this is V. slope is k0. So, we now learn that slope is basically the tilt. So, this tilt is called the slope. So, this tilt, how much this is tilting, this is called the slope. So, the slope is k0, that is the polymerization rate constant. So, once we understand this, that polymerization rate constant is k0, what can we say about other filaments without doing any experiment? We can roughly draw how will the curve look like for some other filament. We drew for actin, we can see how would it look like for another filament. So, let us quickly understand for actin from whatever we learnt. Let us have a look at the values of k0 and kd for actin. So, what we for any polymer, what you can do experimentally is draw velocity versus concentration graph, find the derivative, and this derivatives gives you k0, find the y intercept, this gives you the kd. So, this is the recipe essentially for calculating k0 and kd, and what we are using here is the simplest form of differential calculus. We are using mathematics essentially to learn or to get k0 and kd. Many people unknowingly have used this, but you basically are knowingly use. You have learned about slope, derivative, everything, and now you can understand the whole thing about this and do little more. So, let us look at the actin values. So, now if you look at this for actin, if you look at this curve, what is kd? Kd is the y intercept, this is kd. So, this y intercept here, this gives you kd and this gives you k0. So, now let us look at the kd here. For actin, kd is just below between 1 and 2. So, it turns out that if you look at this point, the point at which this line hits the y axis, or it is called y intercept, kd is turns out to be 1.5 per second, 1.5 per second, and k0, which is the slope of this, is 11 per second, 11 micromolar per second. So, now you can easily look at this. When you how do we calculate the slope without doing too much of calculation? Since this is a straight line, calculating slope is very easy. Look at the change in x here. X changes from 0 to 1 whole 1 micromolar. And how much did the y change? Y changed from 1.5 to this value, which is just below 10, which is actually 9.5. Sorry, yeah, 9.5. So, 1.5 to minus 1.5 to plus 9.5. So, this is about 11. So, this if you count minus 1.5 to this particular point, which is plus 9.5, this turns out to be 11. So, you can see the slope is dV by dC. So, dC is 1 and dV is 9.5 minus 1.5, which is 11. So, you can see that that is how you get the answer 11. So, take this value of 11 and 1.5 and draw yourself a graph which will look like this, and you can learn. Now, from this, the next question you can ask is, what else can you learn from this? From just by knowing this much, what can you say? What can you? So, the one thing about learning theory or mathematics is that you should be able to predict something without doing any experiment, just about thinking about situations, doing some mathematical calculations, you must be able to predict something. So, just by learning what we learned today, what can we predict? We can predict one thing. So, we can say by just listening to this lecture, you can predict one thing. So, the prediction is this. So, we had this curve which is concentration versus velocity for actin, which, so for this particular curve for actin looked something like this. Now, we predict that if you do an experiment for some other polymer, let us say you do an experiment for microtubule, how will the curve look like? So, as you learned here, the k0 depends is basically the derivative, the slope of the curve is the polymerization rate, and the y-intercept is the depolymerization rate. And you can imagine that for microtubule, the rate of polymerization and rate of depolymerization will be some other value. It could be some other value. So, k0 could be something else for microtubule and kd could be something else. So, if you do an experiment for microtubule, you can predict that you will get some other curve which looks like maybe different. So, let us say you could get some curve which look like this, which has a different y-intercept and a different slope. If you do for some other curve, let us say param filament, you might get some curve like this. For some other polymer, you might get a curve like this. So, what we are predicting here, what we can understand, what we are saying here is that depending on the polymer that you choose, depending on the filament you choose, you might get curves with different slopes and different y-intercept. This is something which you could predict just by understanding that the slope is rate of polymerization and the y-intercept is rate of depolymerization. And you can guess that different polymers will have different rate of polymerization and different rate of depolymerization. So, different slope and different y-intercept. So, this much you can say confidently. So, with this we will stop here. To basically to summarize, we learned a few functions and its derivative. We learned the derivative is slope, and we learned 3 formulae. We learned the derivative of x power n, the sum of 2 functions and its derivative, product of 2 functions as derivative. Use that formula to calculate, use those formulae to calculate derivatives of many functions. And one example we discussed now, which is basically the polymerization of actin. And using the ideas of calculus, we learned how to get rate of polymerization and rate of depolymerization, and using that we could say something about how the velocity versus concentration curve will look like for some other filament. So, this much we could, and so with discussing this much, we will stop here, and we will continue with more examples in the coming classes. We will take some other examples and see what we can learn from that example that we did not know. So, the mathematics we learn will help us to understand something more and predict something which we did not know. Okay, so let us stop here. Thank you.